Okay, our next podcast is going to go into inheritance and the study of how genetics and how traits are carried from one generation to the next. We can start by talking about Mendel. Mendel is considered the father of genetics, and he really gave us an understanding of how these traits are passed on. He worked with uh, garden peas is what he worked with, and he would cross these and then pass them on. But he was a monk. This is his fellow monks here, and but really gave us our understanding of what genetics is that we still work with today. There were prior ideas prior to Mendel and his understanding of how these traits are passed on. There was some different ideas. It was always known that certain traits were passed on. You could see those traits in the offspring. Some of the ideas prior to Mendel were kind of a blending hypothesis that the mother and the father, the offspring, would have a blended combination of traits. So that was a common hypothesis, the blending hypothesis. Another hypothesis was something called pangenes, or also sometimes referred to as the particulate hypothesis. And this was the idea that there were these units, they weren't referred to as genes, but there were these units that would carry the particular information for traits, and they were passed on to the next generation um, through the sperm and egg. So there was some truth to it, some accuracy to it, but it was the idea that these pangenes or particulates would travel from that particular trait to the sex organs and then get into the sperm and egg. So if you had brown hair, something from your brown hair would travel to the sex organs and then get into the sperm or egg at that point. Um, and so Mendel showed us that that's not completely accurate because our information for brown hair is in every one of our cells. So it's one of the genes on our DNA, and we carry on our in our chromosomes all of the DNA. So anyway, that was some of the background prior to Mendel. Now Mendel, what he did was he would cross a lot of these pea plants to show who was passing on the trait or not passing on a trait. And so, as I mentioned, he worked with garden peas, and what he would do is cross different varieties. The garden peas have a lot of different um, varieties. Some of the varieties have different flower colors that they produce. Some have different type pods that the peas will develop in. Some grow tall, some are short. So you have all these different varieties, and he would cross these varieties and then track the outcomes of those cross. So this is the technique that he used right here in crossing. So he would first cut off all of the stamens off of one variety. The stamens is what produces the sperm, the male reproductive gametes. And then he would leave the carpel, which is the female accepting the pollen, which carries the sperm. And then he would transfer pollen from one variety to the carpel where the eggs are in another variety. And so what would develop from there is a cross. The sperm would cross with the egg of this variety. In this, we're looking at, for this example, we're crossing the pea plants that produce two different colors, a white color, flower color and a purple flower color. So he would cross those and he would get these peas to develop, which are a hybrid of this, and then he would plant these peas and see what he would get as an outcome for his first generation. We refer to that first generation as the filial generation. And so in this case, every time he crossed it, he did not get a blending. So there was not a blending of purple and white flower colors. So he really kind of showed us that blending is not accurate. But he did not get any white. In the outcome, he got all purple flower color of that variety. So what he ended up figuring out is that there's dominant and recessive. We'll get to that in a minute. But so this is the technique that he used in crossing these different varieties. And so he would track these characters in a very organized way. He would track these in the F1 or the first generation or the F1 generation. Then he could cross some of these with some other, cross some of these together. And then that would be considered our F2 generation. So he would follow this out a few generations to see what the outcome was. And so that gave him a lot of data information as well. Now, the basic unit of heredity that we now know is what was carrying this information is a gene. So a gene is considered the basic unit of heredity. These are found on the chromosomes. It's the chromosomes made up of long segments of DNA, and pieces of that DNA are referred to as genes. Now Mendel recognized some different laws that governed this tracking or passing on of these genes, and he referred to these as the laws of segregation. That is that we get a particular ratio that would come out of these crosses. So during the production of the sperm and egg, you have two chromosomes for each of your set of genes. One set came from your mother and one set came from your father, and then when you produced your sperm and egg in meiosis, then it would split those. And so that pairing would split up, and your gametes would separate out into different sperm or egg, into different gametes. And so this is known as the law of segregation. So the splitting out of those particular traits. Now, the term allele is a word that we use to describe the pairs of those of each member of those genes is referred to as an allele. So we can talk about the allele for purple flower, or the allele for white flower, and so allele is a term that's used for that. So here's a common experiment that we just showed there, but this shows the next generation of crossing that first F1 generation. So this is called our P generation, what we start with there, that true breeding parent that we crossed, and we get all plants have the purple flower color for the first generation. Then we cross some of these, and what Mendel found was that pretty much the ratio was for every 4 that were produced from this cross of the F1 generation, you would get 3 purples and 1 white. And so we found that ratio to be consistent, and 3 to 1 to be a consistent ratio that he found. And so this was showing that what he determined was that some of these were dominant and some were recessive. So the dominant traits would clearly out-express, or they would be expressed over the recessive alleles that would be in that particular DNA. And so the only time you would have a recessive allele expressed, which he found to be this, the white flower color here in this example, is if you had both parents to distribute a recessive allele to their gametes, because this particular individual right here carries one dominant allele from this parent and one recessive allele from this parent. So when it produces its sperm and egg, it will be producing some sperm that has dominant alleles, some that have recessive alleles. So the only time in the outcome of crossing with these individuals is if you get two individuals that distribute a recessive allele, and that's what happens here. So it's kind of like flipping a coin that when you flip a coin, you have a 50-50 chance of getting heads or tails. The same would be true of this allele. You have a 50-50 chance of a dominant allele being passed on in the gametes or a recessive allele. And then also you only have, when you cross these, you have to have two recessive alleles to express in this outcome to get this particular trait. We can also see this dominant and recessive expressed easily in what's called a Punnett square. And so let's look at a Punnett square. Oh, before looking at that, we can see that these alleles are tracked on the chromosome. So here's our homologous pair of chromosomes, and homologous would be the matching chromosomes that we have. One from our mother, one from our father, and then we have the flower color gene that is on a section of that chromosome somewhere. And so when these split up during meiosis, this chromosome will be carrying the purple flower color allele, this one will be carrying the white flower color. So depending on which sperm or egg gets fertilized is dependent upon which gene will be passed on to your offspring. So again, you have two genes or alleles for a particular trait, and so you pass some on to some, you pass one on to some of your gametes, and you pass the other on to others. All right, and so a Punnett square we can track right here. This is called a Punnett square, so we can see that when we cross these, the purple, our example here, we can put these on a grid, and that's what a Punnett square is. And this is a simple Punnett square. We're tracking one trait. And so we put all the particular possibilities. You can have a possibility of a dominant allele, which is represented here by a capital letter, capital P, and you have a recessive allele, which is represented by a lowercase p. So our white flower color is represented by lowercase p, and our dominant allele is purple flower color, capital P. All right, and so when we cross these are all the particular outcomes. We have this sperm can merge with this egg, we get two dominant alleles. This sperm can merge with that egg, we get one dominant, one recessive, and the dominant out expresses, so we will have that expression. In this case, we have dominant merging with a recessive, dominant out expresses, and in this one and only case, we get a recessive merging with a recessive. That's our 1 out of 4. Okay, we're going to stop our podcast here, but we are going to pick up with looking at some variations that occur within Mendel's principles.